note, I've been getting some complaints about the noise level in the room, and I know that in the back especially it's probably fairly difficult to hear, so all I ask is you have a little bit of respect for the people around you, and when I'm going on about something, whether you're interested or not, try to be quiet so other people can hear that might be interested. And if you're really not interested, then leave. I mean, I don't have an attendance policy, so you don't have to be here. So try to keep the noise level down, and I think a lot of people will appreciate it. We need to move into Chapter 3. I've got basically today and next week to cover 2 chapters, so we're going to try to go fairly quickly. This chapter starts to extend the principles of chemistry that we looked at in Chapter 2 to start looking at biological molecules. What kind of molecules do living organisms use? How are they constructed? How are they used? And what are their purposes? Some major points about this chapter. This is really a fundamental statement. Life depends on the properties of carbon. If carbon didn't have the properties it has, if it didn't form 4 bonds, covalent bonds that are really stable bonds, life would not be possible. All right? That's why we anticipate that any other forms of life that we might find on other planets, for example, would be carbon-based life. Because carbon really is the only element up there that has the kind of properties that we really need to look at. The element below it, silicon, sort of has the properties, but its covalent bonds are not as strong. So it would be a much different kind of life. Molecules of living systems interact with each other by using specific portions of those molecules that have specific structure. We call those functional groups, and there's a half a dozen or so of those functional groups that we'll need to take a look at. Okay, those are the regions of the molecule that actually participate in the reactions that are going on. Right? And these are the 4 classes of molecules that we'll look at: carbohydrates, lipids, proteins, and nucleic acids. Those 4 categories of biological molecules are the major categories that we use. Proteins are used for a huge number of functions. Carbohydrates are used mostly for energy storage, but they can be structural molecules too. They can contribute to the structure of the cell. Lipids are energy storage, but they also form the membranes around cells, and so they're very important for that reason. And then, of course, nucleic acids constitute the genetic component of the living organism. So we'll look at each one of these in some detail. All right, the first thing to do is define what an organic molecule is. It's just a molecule that has carbon. Right? That's an organic molecule. Any molecule that has carbon. We sort of fudge a little bit there because we talk about inorganic carbon, like carbon dioxide, we might say is inorganic carbon. But technically, organic chemistry deals with anything that has carbon in it. So, carbon forms 4 covalent bonds, as you've seen, and those are stable bonds. They're not terribly easy to break. It takes a fair amount of energy to do that. And so for that reason, it produces a huge variety of different kinds of molecules. Organic chemistry is a monstrously huge field simply because carbon is so versatile. It can do many, many different things. It can form bonds with itself. It can form bonds with many other kinds of molecules. Right? So there's lots of different organizations and arrangements. And your book has a figure that shows some of these. Okay? Carbon bonds form a tetrahedral shape, as you see in the top of this slide, okay, this figure. We use a structural formula to show the atoms that are present in this molecule as well as the bonds. These lines represent the bonds. But with a ball and stick model, we'd actually have 45-degree angles for these bonds. And then a space-filling model is much more close to what we'd actually see in the real world. You don't have sort of these atoms separated from each other with little sticks representing the bonds. Right? They're sort of stuck together like this with electron clouds around them. Right? So that space-filling model is a much more accurate interpretation. Well, carbon can form skeletons of varying lengths, as is shown in the second part of this figure. Those carbon skeletons can be a few carbons, they can be thousands, they can be millions of carbons. Okay? And those carbon chains are very stable structures. Just if you're curious, the names of these molecules refer to how many carbons there are. This prefix F, that means there's 2, meth means 1, prop means 3, and bute means 4. Okay? And then after that, if you got 5 or more, you name them just according to the Latin number, you'd say, or the Greek number. You'd say pent for 5, hex for 6, hept for 7, etc. Okay, so those carbon skeletons can vary in length. They can be straight chains or they can be branched. All right, here we have a butane molecule, and with 4 carbons, if we have 3 carbons with another carbon branching off, it's actually not butane at all. This is incorrect. Okay, according to organic chemistry conventions, this would be a prop compound because there's 3 carbons in the skeleton. It'd be isopropane, not isobutane. All right, so that's a mistake in your book. But it's still got 4 carbons. You can have single bonds, double bonds, triple bonds. Okay, so, and the location of those bonds can differ. This number tells us where the bond is. All right, so this 1-butene tells us there's a double bond between the first and second carbons. This suffix ene means a double bond. Okay, so the neat thing about the naming conventions in organic chemistry, and if you're curious about those, take organic chemistry. We're not going to mess with them here. But the neat thing about those names is that you can name a compound and know instantly what it looks like. Okay, that was the goal. You don't have to have straight chains of carbons. Carbons can form rings. Okay, they can form baskets of rings. Okay, there's a molecule called Buckminster fullerene that is simply a basket of carbons, and it forms a hollow space inside. Okay, and within those rings, you can have single bonds, as you have in the cyclohexane. The cyclo means that it's a ring of 6 carbons. Benzene has double bonds in the ring. Okay, so there's a wide variety of molecules that you can form using just carbons and hydrogens. Right, never mind adding nitrogens and oxygens in. When you do that, you've got even more diversity. Okay. In all of these molecules, the extra bonds are usually filled in by hydrogens. Right? That's typically the case. You can also break these double bonds and fill them in with hydrogens to make single bonds out of them. Okay, functional groups. These are the specific portions of biomolecules that participate in chemical reactions of one sort or another. All right, carbon skeletons are stable. They can be broken, but it takes some energy to do that. It's not easy to do. So instead of cells trying to break those carbon skeletons, they rearrange molecules by using these functional groups instead. Okay? They almost always contain polar covalent bonds. The reason for that is because polar covalent bonds are easier to break than are regular covalent bonds. Okay? Polar covalent bonds are weaker. Remember water. Water has polar covalent bonds, and water will dissociate spontaneously. Okay? Just on its own. So they require less energy to be broken and reformed, and for that reason they are sort of the best bond to use for these functional groups. You wouldn't want to use ionic bonds because ionic bonds break down in water. Right? And life arose in a watery environment. All right, so what are these functional groups? Well, this table shows most of them, but not all of them. You've already seen the hydroxyl. I talked about hydroxyls when I talked about the dissociation of water. Right up here at the top of this figure is a hydroxyl. The general formula is an OH. We find those in molecules that we generally call alcohols. Okay, so alcohols are defined by the presence of OH groups. Here's an example, ethanol. Okay. This is drinking alcohol. This is the least poisonous of the alcohols. Most other alcohols are very poisonous. Certainly methanol is. All right, we find these in sugars. We find them in water-soluble vitamins that living organisms use. If you look at sugars, as we'll do in a little bit, you find they have lots of hydroxyl groups, and that actually contributes to the sweetness, how we perceive the sweetness of a sugar. The next kind of group is the carbonyl, which is a CO. It's usually a double bond to oxygen. Okay, so a carbon double bonded to an oxygen is a carbonyl. There's two different types, aldehydes and ketones. An aldehyde has a C double bond O with a hydrogen on it. A carbonyl has a C double bond O with something else on either side. Never mind what that something else may be. Okay, as long as it's not a hydrogen, then it's ketone. All right, so here's an example, propanal. That Al on the end, this prop tells us there's 3 carbons. The Al tells us it's an aldehyde. All right, so you can instantly draw the structure of this thing. It's got 3 carbons. The 3rd carbon has an aldehyde on it. Formaldehyde, which is a preservative that's commonly used, is an aldehyde. Some sugars are aldehyde sugars. They have an aldehyde group. We call those aldose sugars. As far as ketones go, we have some sugars that instead have ketones instead of aldehydes on them. We call those ketose sugars. Okay? Here's an example of a molecule that has a ketone in it, acetone. Again, the O-N-E then tells you that that thing has a ketone group. Okay, so aldose sugars, ketose sugars both have carbonyl groups in them. The carboxyl is another important functional group. The carboxyl is COOH, and its structure is like this. A carbon double bonded to an oxygen, and then there's a hydroxyl coming off. Okay, so that together is a carboxyl, or carboxylic acid. It's an acid because this hydrogen tends to drop off in the water, so you get hydrogen ions added to the solution, and when you add hydrogen ions, of course, that lowers the pH. That's why they're acids. So here's an example, acetic acid. Vinegar is, what, 3% acetic acid? And you see that there's that carboxyl group. Amino acids, which are used to make proteins, have carboxyls in them. Some vitamins, fatty acids have carboxyls in them. And we'll look at all those molecules. The amino group is another important one, an NH2. Okay? This thing acts as a base in solution because it tends to pick up hydrogen ions and become charged. So it becomes an NH3+. All right? So it acts as a base. Amino acids have these amines in them. So proteins do as well. Urea, which is a breakdown product of protein metabolism in urine. That's the main reason why you have to pee, is because you have to get rid of that urea. Right? It's a waste product. It's poisonous. All right, so there's 4 of the functional groups, the hydroxyl, carbonyl, carboxyl, and amino, but there's 2 others that we need to look at. The sulfhydryl and the phosphate, right, which your book doesn't talk about as much. Okay. The sulfhydryl is an SH. Okay, so there's a sulfur with a hydrogen on it. And then the phosphate has a phosphorus atom, 4 oxygens, and 2 hydrogens, and then it's connected to something else. Phosphorus is kind of neat. It can form 5 bonds. Right? So it's sort of interesting for that reason. As I note, a lot of these functional groups are ionic. As I said, a carboxyl group will drop off a hydrogen, become negatively charged. An amino group will pick up a hydrogen ion and become positively charged. So when you throw these things in solution, or when you're looking at them in the living cell, they're going to be charged. Okay? They're really no different. Okay, so there's the amino group. Sulfhydryls, you might expect to do this as well, because you might think, well, okay, a sulfur, it's a fairly big atom. It's attached to a hydrogen, which is really little and can't keep those electrons pulled away. So you might think it's a polar covalent bond. It is, but it's only slightly polar. Okay? Because sulfur is actually bigger than oxygen, and it doesn't pull as well. Okay, so the sulfhydryl group is slightly polar. And because of that, it doesn't usually lose that hydrogen. Okay, well, let's see if you got it. Which of the following statements is not true? Okay, try to figure out which one of those is incorrect. Okay, let's go through these one by one and see which ones are true and which ones aren't. What about A? Is that a true statement? Yep, that's a true statement. That's the hallmark of the amino group. It's an NH2. Okay? Organic molecules contain carbon. Obviously a true statement. Right? Sulfhydryl groups are usually not ionic at cellular pH. Well, I just made a big deal about that, so you better remember that they're not. Okay, D, all functional groups contain a carbon atom. Right? Well, I can think of most of them that actually don't. Right? And of course, statement E is true. Hydroxyl groups are found in alcohols. So the answer here must be D. Okay? Only carboxyl and carbonyl groups contain carbon. The rest of the functional groups don't have carbon in them. Right? So this is another one of those kinds of questions that I like to ask where it doesn't just ask for brute force memorization. You have to think about what you've memorized and apply it. And you have to sort of pull it together. Those are the kinds of questions that I really like to ask because they really help to demonstrate that you know what you're doing. Okay, macromolecules. So far we've been talking about fairly simple, small molecules. Macromolecules are big, big aggregates, hundreds or thousands of atoms all together. Okay? Since you've got carbon skeletons that can be branched, unbranched, have double bonds, triple bonds, there's just an amazing variety of molecules that you can produce using just carbon as the basic skeleton of the molecule. Okay? So macromolecules are these monstrous assemblies of atoms. Basically, they're built on carbon skeletons, and they use basic building blocks called monomers. Right? So what cells are going to do is take these smaller molecules, these monomers, and stick them together to form the bigger macromolecules, the polymers. Okay? And that's a large part of what our cells do. To make proteins, for example, we take amino acids, which are the monomer, and we assemble those amino acids together into a chain, and that chain then has specific properties that allow the protein to do what it's supposed to do. The protein is a polymer. All right, how do cells do this? They do a reaction called a dehydration reaction. A dehydration is a removal of water. Okay? So now you start to see why we have these functional groups that have hydrogens and hydroxyls on them. Okay? Because you can take a hydroxyl and a hydrogen and put those together to produce water. When you do that, you've got an open bond here and an open bond here. So you just attach those bonds to each other in a sense. Okay, you produce a new covalent bond, and you've successfully attached that monomer to the existing polymer. Okay, so the dehydration reaction is a removal of water, and almost all macromolecules are assembled by these reactions. You can do the reverse as well. Okay, you can do what's called hydrolysis, or think of it as hydrolysis. Lysis means to break. Okay, so if you think hydrolysis, it literally means water breaking, which is shown on the next slide. Okay, water is added to the polymer. to break out the monomers and restore their original configuration and their original bonds. All right, so cells can assemble monomers into big macromolecules. They can also break them down. If they receive those macromolecules from the environment, they can break them down and use those monomers to make their own macromolecules. All right, that's another major function that cells will do. Okay, this is what you're doing when you're digesting food. You're breaking down those macromolecules into smaller units. Okay, 4 types of macromolecules that we have in cells. And we'll look at each one separately. Carbohydrates, which are made basically of simple sugars. Lipids, various fats and other hydrophobic molecules that our cells use to make membranes and to store energy. Proteins, or polypeptides, those names are not quite equivalent. And I'll explain as we get into polypeptides why they aren't equivalent. You have to be a little careful about your terminology there. And nucleic acids, okay, the genetic material of cells. Okay, so each has its own characteristic monomers. Each has its own characteristic functional groups, and we'll look at those functional groups as we go through this. All right, so let's start with carbohydrates. You can tell a carbohydrate instantly because it always has some variation of the formula CH2O. And that's why we call them carbohydrates. Okay. They have many hydroxyl groups, which contributes to the sweetness. By the way, this is one reason why antifreeze tastes sweet. And it's why it's so dangerous, because it's got the molecules in there. Ethylene glycol. It has lots of hydroxyl groups. You're laughing, but veterinarians always see many, many cases of animal poisoning because of this. When an animal, if you got a leaky radiator, an animal drinks that antifreeze, right, it'll kill them. It'll kill you too. The cure, interestingly, is to get really roaring, stinking drunk. And the reason is because alcohol binds to the same enzyme that ethylene glycol does. So what you're doing is you're blocking the enzyme, and the ethylene glycol just passes through your system. Right? So that's the cure. Okay. Don't try this at home, please. Carbohydrates also have carboxyl or carbonyl groups. Okay? They can have aldehydes or ketones, in which case they are aldose or ketose sugars, depending on what they've got. Okay? The carbon skeletons form rings in solution, so that's another way to be able to tell them. They always look like a ring structure with an oxygen in the ring. All right, now, you're not going to have to memorize these monomeric units, but you are going to have to be able to recognize them. So if on an exam I show you 5 different molecules and ask you which one's a sugar, you should recognize the one that's ringed with an oxygen in the ring. Dead giveaway. Okay? Dead giveaway. These sugars, simple sugars, are called monosaccharides. Okay, that's the monomeric unit that's assembled to form the polymer. Okay, here's a couple of examples, glucose and fructose. Okay, notice the functional groups. Lots of hydroxyls on these things. Okay, every carbon just about has a hydroxyl on it. And that's true of fructose as well. But notice this. Okay, glucose is an aldose because it has an aldehyde group. Whereas fructose, fruit sugar, is a ketose because it has a ketone group instead of a aldehyde group. Okay? So we see the basic characteristics of a typical monosaccharide. Lots of hydroxyls, an aldose, an aldehyde or ketone. Okay? And if you're curious, this OSE ending tells you instantly it's a sugar. Okay? It's another little organic chemistry naming convention. Right? But you never see the linear forms. Right? These things always form rings. It's the most stable conformation or shape of the molecule. Okay, so what they're showing here in this figure is the formation of the ring structure from the linear structure. And what they're showing you is where that oxygen comes from that ends up in the ring. Right? This fifth carbon interacts with the first carbon, right, by a dehydration reaction to produce a bond between the oxygen and the two carbons. Right? Then the sixth oxygen is sticking out by itself here at the top. Right? We will abbreviate these structures usually by this sort of thing. Now, looking at this, you'll see why I said these things are rings with oxygen in the ring. A dead giveaway. Right? There's the oxygen. Here's the ring. Another dead giveaway is all these hydroxyl groups. There's tons of them. Okay? So they're never linear in water. You form a ring. The arrows going both ways suggest that this is reversible, and it is, except in the living organism or in a watery environment. It's never really reversible. These things are so stable, these ring structures are so stable that that's the way they are in the living organism. They may flip very briefly back to a linear form, but they'll pop back into the ring form almost immediately. Okay, so the ring is the typical form. Okay, so here's just something to think about over the weekend, is what would fructose's ring structure look like. It's a ketose instead of a aldose, and so you have to think about where the reaction is going to occur to make the ring. We can take monosaccharides and put them together to produce disaccharides. In this case, we're taking two glucoses, doing a dehydration reaction with them, and making a molecule of maltose, or malt sugar. Okay, so a disaccharide has two simple sugars, or monosaccharides, linked by a covalent bond formed by a dehydration reaction. Okay, so there's the dehydration reaction right there at the top. A hydroxyl comes off of one of them, a hydrogen comes off the other, and we form this bond, a carbon-oxygen-carbon bond that we call a glycosidic bond. Right? There's nothing special about it. It's just the same as any other set of covalent bonds. Right? There's a covalent bond between this carbon and the oxygen, and another covalent bond between the oxygen and this carbon in the next ring molecule. They're just covalent bonds. Right? But when you say glycosidic, that means you're referring specifically to that bond in a sugar molecule. Okay. This particular sugar, just sort of interesting to note, is common in plants. Plants make maltose a lot, especially certain plants. Barley has tons of maltose in it. Okay. Let's try another practice question. All right, sucrose is a disaccharide. Therefore, which of these statements is true about sucrose? Okay, how many people say A? Okay, how many people say B? See, I'm writing too easy questions. I got to make the exam harder. Oh, yeah. You're actually right. B is the correct answer. The reason I asked about A is because you'd see disaccharide and you'd see 2 glucose molecules and you think, oh yeah, okay, 2 molecules, that must be right. It's not. A disaccharide is formed by dehydration of synthesis of 2 monosaccharides. Okay? The monosaccharides that participate in that determine what the disaccharide is. Right? Sucrose, for example, is formed from a molecule of glucose and a molecule of fructose. Maltose, we saw, was formed by 2 molecules of glucose. Okay, so you use different kinds of sugars, and you get different disaccharides out of them that have different properties, particularly different levels of how we perceive their sweetness.
How do you know that it's done by dehydration?
Because the previous slide showed a dehydration reaction. Let me go back to that.
How do you know it's done?
You don't know out of the name. You know because you know that sugars are assembled by dehydration reactions. Okay? And if we look at the previous slide, if I can get there. There we go. Okay? This is standard. This is not specific just to glucose. That's standard for every monosaccharide that is assembled into a disaccharide or polysaccharide. It's a dehydration reaction here that forms that glycosidic bond. Okay? All right, polysaccharides. We go to the next step up. We take lots of monosaccharides and assemble them end to end, maybe branch them a little bit, and we have a polysaccharide made up of thousands or hundreds of thousands even of simple monosaccharide units. Okay, so they're long chains of simple sugars. They may be branched or unbranched, though. Okay, they may contain several different kinds of sugars, although the most common kind of sugar that you see in these polysaccharides, at least in living systems, is glucose. Okay, glucose is sort of the preferred sugar molecule for most organisms. Plants make a fair amount of fructose. They make a fair amount of maltose as well, and a few other sugars. But animals use glucose almost exclusively. Okay? The bonds that form and how they form have a dramatic effect on the properties of the polysaccharide. For example, if the bonds face one direction, we can digest them, and if they face the other direction, we can't. All right? So it's a simple fact of where the bonds are that determines whether we can use the material as a food source or not. And I'll show you examples of that. All right? So, some examples. All right? This is a figure from your book. This is a starch. Okay, we're looking at starch granules in a potato tuber, which is part of the root system. Notice that it's all a bunch of glucose monomers. And all we're really showing here is just the oxygen-containing rings and the glycosidic bonds. The bonds are all facing the same way. Right? It's just a long string. There's no branches involved in this molecule at all. Okay? In fact, starch is a general term for any unbranched polysaccharide made of glucose. Okay? In plants, the starch is called amylose. All right, so starch is a much more general term than you may think. Starches are very easily digested by us. Okay? They're rapidly broken down into simple glucose molecules, so they raise your blood sugar dramatically, okay, quickly. Plants use this to store glucose. It's a simple way of storing glucose as an energy source. Right? So as we'll see in Chapter 5 and 6, we haven't got there yet, but glucose is a very good energy storage molecule, and we use glucose as our primary energy source. We can use some other molecules as well. Right? Glycogen is sort of the equivalent of starch, but for animals. This is how we store glucose in our cells. The liver will store large amounts of this stuff. Our muscle cells store a lot of glycogen. The main difference between glycogen and starch is that glycogen has these complicated branches, right? It makes it easier to pack this stuff, and you can actually pack more glucose molecules into a given volume because this stuff is branched. Okay, so it's a little bit more efficient. So when you're undergoing heavy exercise, for example, this is the first stuff you use. Your muscles start breaking down this glycogen. Your liver starts breaking down its glycogen stores to use. Okay, and then those get rebuilt from glucose that comes back in from your diet, or you can resynthesize glucose from lipids. Okay, so branching allows it to pack better, and more glucose can be stored in a smaller space. It's a much more efficient storage molecule. The third type of polysaccharide that's shown in this figure is cellulose. It's a structural polysaccharide. It's not a storage form for glucose at all. Plant cells use this stuff to make their cell walls. Right? The cell wall of a plant is a rigid structure. It resists pressure, and as a result, it is a very sturdy structure for holding the plant cell shape. Okay, so it's used to build cellular structure. It is unbranched. It's made up of glucose, but there's something really interesting about these bonds, which you may already have noticed. Okay, so what makes it different than starch? If you remember, starch was unbranched, but all of the little oxygens, all the glycosidic bonds, pointed downwards. Here they don't. They alternate down, up, down, up, down, up.
So does that mean it can't digest it?
Absolutely. It's that simple fact that makes it undigestible for us. Okay? We have bacteria in our gut that will digest this stuff. Okay? It'll break it down into smaller molecules and end up producing things like methane out of it. Okay? So that's why a lot of people don't tolerate lots of leafy vegetables, because they produce lots of their bacteria produce lots of methane, bloats them. But it's actually a good thing that those bacteria are there, because it turns out that they break this stuff down into other carbon molecules that have 3 or 4 carbons in the chain, and it turns out that those kind of molecules stimulate our natural anti-cancer proteins. And so, you know, it's been said for a long time that a diet that's high in fiber will protect you from colon cancer. We know now there's a biochemical basis for that. It's because you produce these little 3 and 4 chain carbon skeletons which stimulate your own protective measures, and those protective proteins will kill a cell that becomes cancerous. Right? So it really does protect you. I just don't see how people can get the 30 or 40 grams of fiber a day that the FDA recommends.
So if you just take carbon, you'll get a vitamin from carbon at the store, it will be the same as eating all this broken.
No, it wouldn't, because it would be broken down in your gut. See, that's the problem. You have to have the bacteria in your large intestine actually digesting this stuff and making those little 3 and 4 carbon chains. Okay, so this is insoluble fiber in our diet. We can't eat it. Okay, any questions about polysaccharides before we go on?
Okay, that one, it cannot be digested. You said it can't be digested. But we can eat it, and in our stomach, it breaks it all up and spits it through our large intestines. So it is digestible.
Okay, the question is, it can't, I said it can't be digested, but do our stomachs actually break the stuff down and send it through? No, they don't, as a matter of fact. What happens is that cellulose passes through our stomach and our small intestine undigested. It's once it gets into the large bowel where you actually have bacterial action breaking the stuff down. Okay, and there's some absorption that goes on there, but most of what goes on in the large intestine is absorption of water and salts. So there's really not that much absorption of nutrients that goes on there. Okay? So yeah, we don't have any enzymes at all that can break down cellulose, but the bacteria that live in our guts, our large intestine, can. Okay, any other questions? Okay. On to lipids. Okay, lipids are really loosely defined by your book. They just define them as any sort of hydrocarbon, that is a molecule with carbons and hydrogens in it, that are hydrophobic. They don't dissolve well in water, but they will dissolve in other lipids or oils. Okay. So fats are specifically lipids made from fatty acids and glycerol. Okay, so those are the two components, fatty acids and glycerol, and I'll show you figures of both of these. Phospholipids are a special case. Phospholipids are also made from fatty acids and glycerol, but they have a polar head. In fact, that polar head is charged. It has a phosphate group. It typically has an amino group on it, and both of those are charged in the cell. Okay, so that interacts with water, whereas the rest of the molecule doesn't. Okay, waxes are just a single fatty acid linked to an alcohol. So they're very hydrophobic. And they're typically solid at room temperature. Okay, steroids are just based on cholesterol. They're lipids based on a cholesterol skeleton. And our bodies use steroids for a number of different purposes. All right, so those are the major types. All right, here is the basic units of the fatty or the lipid. Okay, the fat. We have glycerol. The OL tells you that it's an alcohol. And it turns out it has 3 hydroxyl groups on it. Here's a fatty acid. Notice this fatty acid is just a long chain hydrocarbon with a carboxyl on the end. That's where the acid comes from in the name. Okay, fatty acids are organic acids. They have a carboxyl group, and in solution that hydrogen tends to pop off. Okay, when it participates in a reaction like this, however, you get that OH and hydrogen off of this glycerol, and you pull that off in a classic dehydration synthesis. And that produces a bond. Okay, so a dehydration reaction is going to link these molecules together. You take 3 fatty acid chains, and you stick them onto those OHs of the glycerol, and you have a fat. That's a fat molecule. Okay. There is some oxygen in there, which you'd think might make it a little bit polar, but it really isn't. That oxygen is so protected or so sequestered away from the rest of the, or away from interaction with water that it just can't do it. So this is a very hydrophobic molecule. Right? So here is a fat. It's got 3 fatty acids. It's got a glycerol backbone. Notice that those fats, or those fatty acids, are of different lengths. Okay? This one has 1 or 2 more carbons than the top one. Notice also they can have double bonds. Okay? And those double bonds produce a kink in the molecule because of the bond angles that are involved. Remember, the bond angle of a carbon bond is 45 degrees. Okay, what we've done is we've basically bent that a little bit by producing a double bond. And having a double bond in there means that you get a kink. Right? So some of the chains are straight, others are bent. Those double bonds actually make a big difference. If there are no double bonds, they are saturated fats. And if they have double bonds, they are unsaturated fats. You may have looked on a bottle of corn oil or canola oil and noticed that they are polyunsaturated fats. The poly means that they have many chains with double bonds in them. Okay? So I asked, why are unsaturated fats liquid at room temperature while fats, saturated fats, are solid? Animal fats tend to be saturated fats. That's why they're solid at room temperature. Plant oils tend to be unsaturated, and so they're liquid. They're also healthier for you because they're easier to mobilize than animal fats. All right, well, the reason for this is because of this kink that's introduced. If this was a straight chain, okay, then this molecule could pack very closely to another one like it. Whereas if you have a kink in here, it introduces a little bit of a space between this molecule and then the next one that would pack into it. As a result, the kinked ones tend to be looser. They tend to be away from each other a little bit farther than would the straight chain ones. Straight chain ones pack in really well. Because of that, at room temperature, straight chain fatty acids tend to be solids, like animal fats, like lard. The plant oils, because they have the kinks, tend to be unsaturated, or tend to be liquid. Okay? You can make plant oils saturated, however. If you look, a lot of candy wrappers and cookies and that sort of thing will contain partially hydrogenated coconut oil or peanut oil. What that means is that hydrogens, the stuff has been reacted with hydrogen, and that's broken the double bonds and made them single bonds so that they're straight chain. It makes the oils thicker, and they're easier to use for that baking purpose. Right? But they're not as healthy for you. In fact, I've been told, and I don't know if this is true, I've been told that partially hydrogenated coconut oil is about the worst thing you can possibly eat. Okay, so that explains the physical properties of these molecules. This stuff is cholesterol. Right? We get a lot of cholesterol from our diet. The amazing thing is we don't need a bit of it. Right? Our liver is very adept at making cholesterol. There's no reason we should have to get it from our diet. And so what we tend to do is we tend to store it, circulate it around in our blood supply, and if we're not able to mobilize it very well, we end up clogging our arteries with the stuff. We do use it, however, for making things like testosterone, estrogens, progesterone, a whole bunch of other hormones, glucocorticoids, mineralocorticoids. Those are all involved in salt and water balance and energy use. And of course, estrogen, progesterone, and testosterone are sex-specific hormones. Okay, so we do make small amounts of those hormones using cholesterol. Cholesterol is an integral part of the cell membrane. But again, we don't need any from our diet. We make all of it that we need ourselves. Okay, so, lowering your cholesterol is not going to do you any harm. All right. Which of the following would be most hydrophilic? Which of the following would interact with water the best? Okay, well, these are all pretty hydrophobic molecules. Waxes are a fatty acid with an alcohol, really hydrophobic. Cholesterol is very hydrophobic. Saturated and unsaturated fats are very hydrophobic as well. For that matter, so are phospholipids, but phospholipids have one fundamental difference. They have a polar head. They have a phosphate group, typically a nitrogen group. Those are charged, and those interact with water. So of this list, the one that would interact with water the best would be D. Okay? The degree of saturation really has little to no effect on how well they interact with water. It all has to do with polar bonds. Okay? All right, we'll stop there, and we'll just continue from this point on Monday, since you're all so darn antsy.
I used to volunteer